Hey everyone, welcome to this deep dive, and it's made especially for all of you in Mr. Martin's IB biology class.
That's right.
We're going to be talking about enzymes and metabolism.
Yeah.
And, it might not sound super exciting at first, but trust me,
Oh, it is.
it is really fascinating stuff.
It's amazing.
Um, think about it this way. Like when you're working out really hard and your muscles are sore the next day,
Yeah.
that recovery process, how your muscles rebuild and repair themselves,
Uh-huh.
that's all thanks to enzymes.
Yeah, and even like when you get a cut,
Right.
the healing process, how
Yeah.
quickly it closes up, or just you know how our bodies get rid of waste efficiently?
Right.
It's all because of these little things called enzymes.
So basically, without enzymes, we'd be in big trouble.
Oh yeah, we would be either moving super slow, like slugs, literally, or we'd have to be incredibly hot, like temperature hot, to function properly.
And neither of those options sounds very appealing.
Not at all.
So, all right, let's break this down a little bit.
Okay.
What exactly are enzymes, and how do they fit into this bigger picture of metabolism?
Well, enzymes are like these tiny little machines that are found inside our cells, and they make all the chemical reactions in our bodies happen like way faster.
So they're like little catalysts speeding things up.
Exactly. And the amazing thing is that each enzyme is super specific about what it works on.
Oh, so like lactase.
Yeah.
The enzyme that breaks down lactose can only break down lactose.
That's a perfect example. So it's like each enzyme has its own special job to do.
So how do they know what to do? How do they work?
It all comes down to their structure. You see, enzymes are proteins, and they have this really unique 3D structure that's kind of like a custom designed tool for a specific job.
So it's like a lock and key kind of thing.
Yeah, that's a good analogy, but it's actually a bit more dynamic than that.
What do you mean?
Well, it's called the induced fit model, and it suggests that the active site, which is the part of the enzyme that binds to the reactant, or what we call the substrate,
Okay.
it actually changes shape slightly to perfectly fit the substrate.
Oh, so it's not just a rigid fit.
Right, it's more like putting on a glove. You know how your hand kind of shapes the glove for a snug fit?
Yeah.
It's similar to that. The substrate interacts with the active site, and they kind of mold to each other to optimize the enzyme's function.
That's really cool. So that slight adjustment is what makes the enzyme so efficient.
Exactly. It's like having all the right tools perfectly aligned to build something quickly and effectively.
So we've got these precisely shaped enzymes speeding up reactions all over our bodies. And all those reactions together make up what we call metabolism.
That's right. Metabolism is like the sum total of all the building and breaking down processes happening inside your cells right now.
So it's like this huge network of reactions happening all the time.
Yeah, and all of it is carefully coordinated by those remarkable enzymes we were just talking about.
Okay, so let's zoom out a little bit and look at this bigger picture of metabolism.
Okay.
What are the main types of reactions that are happening in our cells?
Well, we can divide metabolism into two main categories, anabolism and catabolism.
Okay, I've heard those terms before, but I always get them mixed up.
Yeah, it's easy to do that. So think of anabolism as the building up processes.
So taking smaller molecules and putting them together to make bigger ones.
Exactly. It's like building a complex Lego masterpiece piece by piece. You're taking simple building blocks and creating something more elaborate.
I like that analogy.
Good. So some examples of anabolic reactions are things like protein synthesis, where amino acids are linked together to form proteins, or photosynthesis, where plants use sunlight, water, and carbon dioxide to build sugars.
So those processes require energy, right?
Yes, anabolic reactions need an input of energy to drive those building processes, just like you need energy to put those Lego pieces together.
That makes sense. And what about catabolism?
Catabolism is the opposite. It's all about breaking things down.
Oh, so like taking that Lego masterpiece and dismantling it.
Exactly. You're breaking down those complex molecules back into their smaller building blocks.
Okay, so some examples of that would be digestion.
Exactly. When we eat food, our bodies break down those large food molecules into simpler ones that our cells can use, and then there's cellular respiration.
Which is how our cells extract energy from glucose.
You got it. And these catabolic reactions release energy. It's like when you knock over that carefully built Lego tower and everything goes flying.
And that released energy is what our cells use to power other processes.
Exactly. It's a beautiful cycle of building up and breaking down, and both of these processes, anabolism and catabolism, rely on enzymes to make sure everything runs smoothly.
So we've got these tiny machines, the enzymes working within this incredible network of reactions we call metabolism.
It's pretty mind-blowing when you think about it.
It really is. But how do our cells make sure everything is running at the right pace? I mean, are enzymes always on?
That's a great question. It's like having a car with only an accelerator, you need brakes too, right?
Right, otherwise you'd be crashing all the time.
Exactly. So enzymes are incredibly efficient, but they also need to be controlled so that our cells don't run out of control.
So what are some of the ways that our cells can regulate these enzymatic speed demons?
Well, for starters, enzymes are quite sensitive to their surroundings. They have specific conditions where they work best. It's kind of like a Goldilocks situation.
Not too hot, not too cold, but just right.
Precisely, and one of the most important factors is temperature.
Oh yeah, because if it's too cold, everything slows down.
Right, the molecules move more slowly, so they bump into each other less frequently, which means the reactions happen more slowly.
And what happens if it gets too hot?
If the temperature gets too high, the enzymes can actually lose their shape.
Oh, so they like melt?
Not exactly melt, but it's called denaturation. That precise 3D structure we talked about is essential for their function. Imagine taking a perfectly folded protein and scrambling it up like a tangled headphone cord.
Oh, I see. So it can't do its job properly anymore.
Exactly. High temperatures disrupt the bonds holding the protein together, and it loses its specific shape and therefore its ability to function.
So temperature is like this Goldilocks zone for enzymes. What about pH? Does that play a role too?
Absolutely. Just like with temperature, enzymes have an optimal pH range where they work best. If the environment becomes too acidic or too basic, it can also lead to denaturation, disrupting the enzyme's structure and function.
It's amazing how sensitive these tiny machines are.
They really are, and there's another factor that comes into play, substrate concentration.
Okay, substrate concentration, what's that?
It basically means how much of the reactant the substrate is available for the enzyme to work on.
So it's like having more ingredients for the enzyme to work with.
Exactly, and the more substrate molecules you have available, the faster the enzyme can work.
Because there are more opportunities for those molecules to bump into the active site.
Right, but there's a limit. Imagine you have a team of workers assembling those Lego models, and you keep giving them more and more pieces.
At some point, they can only work so fast, even with all the extra pieces.
Exactly. Enzymes have a maximum speed they can reach once all the active sites are occupied. Adding more substrate won't make the reaction go any faster. It's like a bottleneck on a highway. The cars can only move so fast no matter how many more you add to the road.
So there's a speed limit for enzymes based on these different factors.
That's a great way to put it.
But what happens when our cells need to slow things down or even stop a specific reaction altogether? How do they do that?
Well, that brings us to the fascinating world of enzyme inhibition, which is how our cells can actually put the brakes on these reactions when needed.
Okay. I'm definitely intrigued by that. So tell me more about enzyme inhibition. How does it work?
Well, there are actually several different ways that our cells can inhibit enzymes, and we can categorize them based on how they interact with the enzyme. But we'll dive into all those details after a quick break.
All right, sounds good. We'll be right back. Okay, so we're back and ready to learn more about enzyme inhibition. I'm really curious about how our cells can actually put the brakes on these reactions when they need to.
Yeah, it's a really crucial part of how our cells regulate everything, making sure everything stays balanced and working together properly. Like imagine an orchestra.
Sure.
You've got all these different instruments playing, right? And they all need to be playing at the right volume and the right tempo to make the music sound good.
Right.
So enzyme inhibition is kind of like the conductor controlling the pace and the intensity of all those individual reactions to make sure everything works together in harmony.
I love that analogy. So how do our cells actually do this? How do they put the brakes on these enzymes?
There are a few different ways, and we can group them based on how they interact with the enzyme.
Yeah.
So let's start with competitive inhibition. That one's probably the easiest to understand.
Competitive inhibition. So is it like a competition for the enzyme's attention?
Exactly. You've got this inhibitor molecule that looks a lot like the substrate, the molecule that the enzyme normally binds to.
Uh-huh.
And this inhibitor basically tries to steal the substrate spot. It competes with the substrate for the active site on the enzyme.
So it's like they're both trying to sit in the same chair.
Yeah, that's a good way to think about it. And when the inhibitor binds to the active site, it blocks the substrate from binding, which basically stops the enzyme from doing its job.
So it's like putting a roadblock on the enzyme's highway.
Exactly. And what's interesting is that if you have more substrate around, you can actually overcome this inhibition to some extent.
That's like a numbers game.
Yeah, it's a tug of war between the substrate and the inhibitor. Whoever's more abundant wins.
That makes sense. What are the other types of inhibition? How do they work?
Okay, so let's move on to non-competitive inhibition. This one's a little more complex.
Okay.
In this case, the inhibitor doesn't bind to the active site directly. Instead, it binds to a different site on the enzyme called the allosteric site.
So it's not a direct competition for the active site.
Right, but by binding to this other site, the inhibitor can still mess things up.
How?
Well, enzymes aren't these rigid structures, they're actually quite flexible.
Oh.
So when the inhibitor binds to the allosteric site, it can actually change the shape of the enzyme.
Oh, I see. So even if the substrate can still bind, the active site might not work properly anymore.
Exactly. That change in shape can either make it harder for the substrate to bind, or it can even completely block the active site. And unlike competitive inhibition, increasing the substrate concentration won't overcome this type of inhibition.
That's pretty sneaky. So we've got these two main types of inhibition where the inhibitor can bind and unbind from the enzyme.
Yeah.
What about those irreversible inhibitors we talked about earlier, the ones that are more permanent?
Ah, yes, those are the heavy hitters. Irreversible inhibitors form a really strong bond with the enzyme, like a permanent roadblock. They often target a really important amino acid in the active site, basically changing the enzyme structure permanently and making it inactive.
Wow, so it's like completely breaking the enzyme?
Pretty much. And one of the most famous examples of this is penicillin.
Penicillin, the antibiotic.
Yep. Penicillin works by irreversibly inhibiting an enzyme called transpeptidase, which bacteria need to build their cell walls.
Oh, so it's like taking away the tools they need to build their houses.
Exactly. That's why it's so effective against bacterial infections.
But doesn't that mean it could also harm our own cells?
That's a good question, but luckily penicillin specifically targets an enzyme that's only found in bacteria. Our cells don't have it, so they're safe. It's a great example of how understanding the specific structures and functions of enzymes can help us develop drugs that can target harmful pathogens without harming our own cells.
That's incredible. So we've talked a lot about how our cells can regulate enzymes through different types of inhibition, but I'm wondering, we've talked about breaking things down, but where does the energy come from to build things up? Where does all that cellular energy come from?
That's a great question, and it leads us to the heart of cellular energy production, ATP, and cellular respiration.
Okay, ATP, I've heard of that, but I don't really know what it is.
ATP stands for adenosine triphosphate, and it's basically the energy currency of our cells. Think of it like a tiny rechargeable battery that powers all sorts of cellular work, everything from muscle contractions to nerve impulses to building complex molecules.
So it's like the fuel that keeps everything running. But where do our bodies get all this ATP?
That's where cellular respiration comes in. It's an amazing process that converts the energy stored in the food we eat into this usable form of ATP. So it's like a complex energy conversion system where glucose, a simple sugar from the food we break down, is transformed into ATP, the energy our cells can actually spend.
It's like our bodies have their own little power plant.
Exactly. And there are actually two main pathways for this energy conversion: anaerobic respiration and aerobic respiration. Anaerobic respiration doesn't need oxygen, and it's like a quick burst of energy, great for short, intense activities.
Like if you had to sprint to catch a bus.
Exactly. You can only do it for a short amount of time before you need to catch your breath. Aerobic respiration, on the other hand, needs oxygen, and it's much more efficient at producing ATP, so it's better for sustained activities.
Like going for a long run.
Exactly. Aerobic respiration is our main energy source happening all the time in our cells to provide a steady supply of ATP to keep everything functioning properly.
Okay, so let's break down this aerobic respiration process. Where does it all start?
It all begins in the cytoplasm, the jelly-like stuff inside our cells, with a process called glycolysis.
Glycolysis.
Yeah, during glycolysis, a glucose molecule is split into two smaller molecules called pyruvate, and a little bit of ATP is made in the process.
So we're already making some ATP even before oxygen gets involved. What happens to those pyruvate molecules?
They take a trip to the mitochondria, the powerhouses of the cell. Inside the mitochondria, the pyruvate molecules go through a series of transformations in the Krebs cycle, which makes more ATP and releases high-energy electrons.
High-energy electrons. What do those do?
They're crucial for the final stage of aerobic respiration, the electron transport chain. This chain is located on the inner membrane of the mitochondria, and it's kind of like a cellular conveyor belt passing those high-energy electrons down a line of protein complexes.
And this movement of electrons is what ultimately makes energy.
Exactly. As the electrons move down the chain, their energy is used to pump protons, which are hydrogen ions, across the mitochondrial membrane. And this creates a proton gradient, a difference in concentration between the two sides of the membrane.
So it's like building up potential energy like water behind a dam.
Exactly. And just like water rushing through a dam can generate power, those protons flowing back across the membrane through a special enzyme called ATP synthase generate a ton of ATP.
Wow, so this is where most of the ATP is made during aerobic respiration.
You got it. It's the big payoff. And while aerobic respiration is much more efficient than anaerobic respiration, it's not perfect. Some energy is still lost as heat during these conversions, but even that heat is useful. Our bodies use it to maintain a stable internal temperature.
So it's all connected. It's amazing how efficient and interconnected these processes are.
It really is a testament to the brilliance of nature.
This deep dive into enzymes and metabolism has been really eye-opening, from tiny enzymes speeding up reactions to the huge network of energy production. It's clear that these processes are essential for life.
I completely agree. Understanding these fundamental concepts gives us a much deeper appreciation for the complexity and the beauty of biology.
Absolutely. And a huge thank you to you, our expert, for guiding us through this fascinating journey. We hope you've all enjoyed this deep dive into the world of enzymes and metabolism. And remember, keep those brains buzzing and keep exploring the amazing world of biology. This deep dive into enzymes and metabolism has been so fascinating. Learning about these tiny enzymes speeding up reactions and this whole energy production process has really shown me how complex and amazing our bodies are.
I agree. It's mind-blowing when you think about it, and it really highlights how important these processes are for life.
Absolutely. Well, a huge thank you to you, our expert, for taking us on this incredible journey, and to all of you listening in Mr. Martin's class. We hope you enjoyed this deep dive into the world of enzymes and metabolism.